Hello, everyone. Thank you for joining us today for our webinar, USP Methods for Microbial Contamination in Supplements, CBD Hemp, and Probiotics. This webinar will include a panel of speakers today. The first is Kit Goldman of USP. She is the Director of Dietary Supplements and Herbal Medicines. She will also be joined by her colleague, Radha Tiramalia, Principal Scientific Liaison. And then finally, the last portion of the presentation will be by Shannon Jacoby, Business Unit Manager at Eurofins Microbiology. I am Amy Knock, and I will be moderating today's session. Before we begin, I wanted to just let you know how this webinar will run. This webinar will be recorded, and the slides and recording will be available for you within 3 business days. A short Q&A session will follow the presentation to answer viewer-submitted questions. During this webinar, at any time, you can submit your questions you have using the webinar sidebar menu. Select the Questions tab, type in your question, then hit the Enter key on your keyboard. Remember, you can submit questions at any time. And again, at this point, I would like to turn it over to Kit to begin your presentation. Kit, you may go ahead.
Thank you, Amy. And thank you to Eurofins for arranging this seminar. What we're going to talk about today are microbiology, USP, the general chapters, and the dietary supplements. And the slides won't move. Okay. Let's see here. There we go. All right. We're going to do an introduction to USP, talk a little bit about utility of USP standards, how the documentary standards work together, and then Radha is going to discuss the microbiology chapters, suitability testing, and the new chapter 60. I wanted to do a bit of an introduction to USP because when I was in industry, I knew what USP standards were. I knew when the methods were outdated and annoying, and I knew when there weren't USP ingredients available. But I really didn't understand what USP as an organization was, and I also didn't really understand how the standards worked. So I thought I'd take a few minutes to sort of introduce that. USP turned 200 years old this year. We were chartered in 1825 by Congress. We're a nonprofit organization with the mission to improve global health through public standards and related programs. And that is intended to ensure the quality, safety, and benefit of medicines and foods. We produce different compendia. The main one is the USP NF. These used to be big books. They are now predominantly online. The USP NF contains all of the monographs for APIs, drug products, excipients, dietary supplements, and dietary ingredients used in dietary supplements. We also have the Food Chemicals Codex, the USAN, which is the Dictionary of Terms, and the Dietary Supplements Compendium. USP creates public standards, and we do that with various processes. But we run with expert committees, and the expert committees actually provide input and ballot on all of the documentary standards. The process of getting to that ballot is also a public process where we publish all of the draft chapters and monographs online in what's called the Pharmacopoeia Forum. This is a free forum, and we encourage people to go online, look at the new standards, and make comments. Does this make sense? Definition of this make sense? Does the method work? Are there any other issues or questions that we should know about? And then we take all of that public input into account when we are finalizing the standards. We also create reference standards, which are physical standards related to the monographs. And those support identification testing, related compounds and impurities, limit tests, residual solvents, dissolution, and of course, assay. USP has approximately 9,000 reference standards. And why is it important that these are public standards? They provide a scientific basis for decision making. They ensure a consistent approach to the quality of the products. And they're also very helpful in monitoring for counterfeit, substandard, adulterated products. And right now, with the supply chain so disrupted from COVID, those attributes are more important than ever. And these work through the use of appropriate titles, definitions, validated methods, and established limits. For dietary supplements and herbal medicines, they're very important for industry, for the ingredient manufacturers and the dietary supplement manufacturers. Government regulators respect the quality of the USP standards, whether or not they are required for compliance. And the use of the USP standards gives confidence to practitioners that they're helping their patients select a quality product. When you are a manufacturer, ingredient, or finished product, there are some significant advantages to complying with USP. For one thing, in terms of supply chain transparency, the ingredient supplier and purchaser both understand the quality of the materials purchased. They know exactly what they are getting. They know what the required tests are. And when there is a dispute about whether something meets the specification or not, they know which test is the determining test to use. Also, ingredients that conform to USP that are in a finished product, they may indicate USP in the ingredient table next to the name. And that's an indicator of the quality of the ingredient that was used. So now I'm going to talk about exactly how the documentary standards that USP creates work together. There are 3 types of standards: general notices and requirements, general chapters, which we'll be focusing on today, and individual monographs. General notices were described to me when I started at USP as the 12 most important pages in the compendium. They present all of the basic assumptions about compliance, how to do certain things, conditions for interpretation of data, and they apply to everything in the USPNF. So that's general chapters and monographs, except if it is specifically stated otherwise, and there are some cases where an exception needs to be made. And if that is the case, it will be stated in the general chapter or monograph, and that will then supersede the requirement. In monographs, in addition, there may be a statement that indicates that certain parts of a general chapter are not applicable. So what are the general chapters? They provide guidelines on the activities related to tests and procedures in monographs. And there are 3 different types of general chapters. Numbers 1 through 999 are general tests and assays. They provide specific information about how to conduct a particular test. 1000 through 1999 are general information. That may be background information, may give information on how to validate, may give information about certain equipment or how to select a test. Those are not required chapters. Whereas if 1 to 999 are cited in a monograph, they are required to be followed. Dietary supplements have their own set of general chapters, and those are numbered above 2000. That doesn't mean that dietary supplements may not cite chapter numbers below 2000. It just means that there are additional chapters applicable only to dietary supplements. So what is in the monographs themselves? Those are the descriptions, the definitions for the individual either product or ingredient. So it lists the official and validated tests and the actual test procedures, as well as the acceptance criteria. And together that defines the specifications for the individual product. So that would be identity, purity or limit for contaminants, content, and quality if that is required. And when you think about dietary supplements, that is a very, very broad range of types of products, from relatively straightforward small molecules like vitamins, minerals, amino acids. Then you have proteins, probiotics, botanical extracts, and other types of products. It's a very, very broad range. So the monographs for the dietary supplements can vary based on the type of product that is involved. So when you are working with these general chapters, as I said, if a monograph cites a chapter and it's a monograph for an API or drug product in the US, you must follow the general chapter. Informational chapters are exactly that. They provide information only. For compliance with USP for dietary supplements in the US, that's voluntary. However, if you choose to comply, then if a general chapter is cited in the monograph or general notices, you must follow that general chapter. And now I am going to turn this over to my colleague Radha, who is the Principal Scientific Liaison and works with the Microbiology Expert Committee to discuss the compendial microbiology tests. Radha?
Thank you, Kit. Thank you very much. And thanks for the opportunity from Eurofins to be able to talk to you briefly today to give you some background and be able to answer some of your questions. So let's begin with what does USP does in the area of microbiology? What is its role in microbiology? We heard from Kit about the mission of USP, which is good quality medicines, foods, dietary supplements for all. Compatible with USP's overall mission, the role of USP in microbiology is to develop public standards pertaining to microbiology, be it sterile products or non-sterile products, that along with other requirements will ensure the consistent quality of products, whether it is a dosage form or a drug substance or an excipient or a food ingredient, a dietary supplement, etc. So that is USP develops public standards, as Kit talked about, alluded to public standards and the value of public standards. And that is what we do in the area of microbiology. We have an expert committee that is a dedicated group of expert volunteers that are involved in the area of microbiology and sterility assurance. And I'm glad to report for this cycle, we have a representative from Eurofins. Actually, her name is Herlyn Clow. She's a new addition to our expert committee beginning this cycle. Next slide, Kate. Yeah, this slide lists commonly used USP microbiology general chapters, especially in the areas of non-sterile products including dietary supplements. And Kit explained to you the difference between general chapters numbered less than 1,000 and chapters numbered over 1,000, as well as general chapters numbered over 2,000. So the ones listed here typically apply, as I said, to non-sterile products. And we will talk briefly today. I know there is a presentation from Eurofins on antimicrobial effectiveness testing. We will hear about it and we'll answer any questions that you may have. We will briefly look at differences between 61 and 62 and 2021 and 2022. In addition to these chapters in the area of non-sterile products, we also have chapter 1111, which is a mirror image, you can say, of chapter 2023, and 1111 contains recommended acceptance criteria for products that don't have USP monographs. Okay, next slide. So we'll briefly look at quality of non-sterile products. And regardless of whether a product is sterile or non-sterile, we have to take very careful to make sure that they are within a prescribed quality. And I will mention this why we have to worry about quality of non-sterile products, even though by definition non-sterile products can contain microorganisms. But it's important they do not contain any number of microorganisms. Microorganisms should not be beyond a certain number, as well as the type and the type of microorganisms. Okay, so that is why we look at two different aspects of microbial quality, one related to microbial enumeration, and that is covered for non-sterile pharmaceutical products under USP 61, and for nutritional and dietary supplements under USP 2021. That will give us how many microorganisms are there in a product, and as well as what kind of microorganisms should not be there in your product. And we will come to the question in a minute, why certain microorganisms should not be there. Two reasons here. First thing, it has to do with the number of microorganisms has to do with the quality of the product, the effect of the microorganisms on the quality of the product itself. It should not destroy your product's activity. The second thing is the type of microorganism should not have an effect on the patient or the consumer who is taking these products. So there are two types of effects microorganisms have on your, one is directly on your product. The second thing is on the health of the patient or the consumer. So that is why we have two aspects of quality, one related to the number of microorganisms as well as type of microorganisms. The next slide. Okay, 61, 62, 2021, 2022 are very similar in their approaches and the final goal, microbial quality. So why do we need these two sets of chapters, and what are some differences in these sets of chapters? 61 and 62 apply to non-sterile pharmaceutical products, and they were written subsequent to the development of 2021-2022. Prior to 2006, USP had one chapter, USP 61, that addressed both enumeration as well as the test for absence of certain specific microorganisms. So those were there prior to 2006. And the tests for quality of dietary supplements were actually developed around 2002, 2003, prior to the development of 61 and 62. Although we did have idea at that time that USP 61 is going to split into two chapters. That's why we wrote two separate chapters, one addressing enumeration as well as the second one addressing absence of specific organisms for dietary supplements. And very at a 10,000 foot view, what are the differences between 61, 61, 62 and 2021, 2022? 2021, 2022 are a lot less prescriptive than 61 and 62. They give you more options compared to 61 and 62, less rigid, and as I said, more options in terms of media, how you interpret results, how much challenge organisms it use for suitability, and so on. We will go into details in the table, but the couple of differences right away. For example, the preparatory testing indication in 2021, 2022 is identified as suitability or verification of the counting method in case of 61 and 62. 61 and 62 do not have an option for retest, okay? Instead, there is an option for using the maximum acceptable count because microbiological methods are highly variable. 2021, 2022, on the other hand, have an option for retest using a larger sample size. They do not have the option of maximum acceptable count. So those are some differences. There are advantages and disadvantages because if you have to enact if you want to retest, right now, what you have to do is you have to invalidate your test. To invalidate a test, you have to find out the root cause of the variability or the deviation and be able to pinpoint the root cause prior to retesting. And let's move on and look at some of the differences in the next slide. So the challenge organisms in 61 and 62 are different, okay? For example, we have the use of Candida albicans and Aspergillus brasiliensis is included for total aerobic microbial count, whereas, and also E. coli in 2021 versus Pseudomonas aeruginosa in 61. Similarly, for total aerobic microbial count, the organisms are very similar. In addition, since 2021 also addresses E. coli testing, Salmonella species testing, and also the Enterobacteriaceae testing, we have those challenge organisms indicated in 2021. The inoculum size, okay, the difference here is in case of 61, we say less than 100 colony forming units, whereas in 2021, we say 25 to 250. In practice, there is not much difference because 100 CFU is smack in the middle of the linear range of recovery of microorganisms, which is actually between 25 and 250. We specifically indicate less than 100 colony forming units in 61 because we have a requirement that says that the recovery of the challenge organism should be within a factor of 2. And we don't want that beyond the linear range of the curve. That's why we specifically restrict the challenge organisms to 100. And we don't have that requirement of factor of 2 in 2021. We say greater than 70%. So that is the difference in terms of inoculum for challenge organisms as well as recovery requirements for challenge organisms. Some additional differences in terms of incubation times. And as I said, retest option, okay? So those are some additional differences between 61 and 2021. Next slide. In addition, for 62 versus 2022, again, we have differences in inoculum size. We have the same bile-tolerant gram-negative test, which has both an option to do a qualitative test as well as a quantitative test using a most probable number method. Salmonella species testing requires 10 ml. Media differences are there. You have a restriction only to RVS medium in case of 62, whereas you have additional media options, as I said, more flexible in 2022. Pseudomonas aeruginosa is not a concern in case of dietary supplement products, so we don't have that test in 2022. Staph aureus is the same as 61, but with additional options. No requirement for Candida albicans, which is not a risk involved with dietary supplements, but especially with certain products, pharmaceutical products which need C. albicans testing. Again, the difference with the retest option. The next slide. We will come to address some specific questions that were asked. Why do we need to do method suitability prior to a actual product test in the area of microbiology? Method suitability is nothing but your test for removing any bacteriostasis, fungistasis properties that may be in your products. Because the tests that we do are growth-based microbiology tests. In other words, any microorganism that is present in your product, you should be able to recover that. But if your product has antimicrobial properties such as bactericidal, bacteriostatic, fungicidal, fungistatic, you will have to neutralize that. And that is what we do when we say method suitability. So unless you do that, you cannot have a valid test. And this is consistent with what the FDA says as well, because according to 21 CFR 211-194, it says the method used for testing the sample, the method will meet proper standards of accuracy and reliability as applied to the product tested. Okay, so that is what we're doing. We're making sure that there is nothing in your product and your method is suitable for your product. So you don't have to validate the method itself. You actually make sure that the method is suitable for testing with your product. Next slide. Kit, can we move to the next slide?
I'm trying. It does not wish
Oh.
to move to the next slide again.
Okay.
Let's see. There we go.
Okay. So that is again, that is indicated here as well. Methods that are official in the USP, chapters 61, 51, 62, 71, etc., are considered to be already validated. If microorganisms are added to the test during suitability, it is necessary for you to eliminate, and if you have to recover those things, it is necessary you have to eliminate the biostatic, which is fungistatic and bacteriostatic properties of the product of the article, and guidance on how to do this neutralization or method suitability is given. If, for example, it is inbuilt in 61 and 62, but in case of 2021, 2022, you can actually use the guidance in USP 1227. It provides some overview of the common methods for neutralization, such as chemical neutralization, dilution, filtration, and washing, combination of these things. Next slide. Okay, we will move on to the newly developed chapter, which became official in December of last year, which is microbiological examination non-sterile products test for the Burkholderia cepacia complex. And these tests apply specifically to aqueous-based products because, as you know very well, the source of BCC is going to be the water that you use in the preparation of these products. And these tests apply specifically to aqueous products, particularly those for inhalation use or aqueous preparations for oral, oromucosal, cutaneous, or nasal use. And as you all know, BCC is a complex of 23 members there in their family. And this was necessitated, next slide, because the FDA issued an advisory in 2017 to manufacturers of aqueous non-sterile products based on several product recalls and health issues resulting from BCC contamination of non-sterile aqueous products. FDA issued an advisory that manufacturers of aqueous non-sterile products have to control for the presence of BCC. Unfortunately, at that time, there was no standard method available in any regulatory or in any of the pharmacopoeias. So at that time, FDA said use scientifically sound appropriate acceptance criteria, such as in USP 1111, following procedures similar to USP 61 and 62, to assure that drug product components also, including water, pharmaceutical water, and finished drug products, conform to appropriate quality standards. Next slide. And in 2018, they further went on to say, this is from a presentation by Stephen Langell at the FDA conference, why it is important to control and review the trending of microbiological water quality, especially because BCC members have this unique property of overcoming the effectiveness of preservatives. So even if you use preservatives, there is no guarantee that BCC will not grow in such systems. So that is the reason why FDA asks manufacturers of non-sterile products, aqueous products specifically, to pay close attention and monitor for the presence of BCC members. Next slide. So at that time, FDA USP worked with based on stakeholder request to develop a chapter for BCC, that is USP 60. So we worked with the FDA labs as well as Health Canada labs to develop validated methods for detecting the presence of BCC. And although a couple of options were available based on the validation data that we saw, we suggested the use of BCSA, which is B-sepacious selective agar, for the use as a diagnostic medium. It has been widely used in clinical microbiology, especially. And we also suggested the use of 3 species from the BCC members, the 23 members. These 3 were the ones that are mostly associated patient infection. So you see this in clinical settings. So those were this, we suggested 3 and not 1 at this time because we wanted to gather data of whether you need to test for more than 1 member or more than 1 species. So as data becomes available, we certainly have the intention of reducing the number of species that you need to use for growth promotion, as well as method suitability and BCSA from 3 to reduce it certainly to 2 and probably 1. And the other indication that we have suggested is the use of diluted soybean casein digest broth, especially for the first step or the general enrichment step, especially when you use when you're trying to test for BCC in water, in purified water. BCC, especially in from purified water, does not adapt very well to high strength the soybean casein digest medium. So you can use diluted, such as 1 to 10 diluted soybean casein digest broth for the general enrichment step, especially for those types of products. We did not make this a part of the harmonized chapter 62, since the other pharmacopoeias don't have corresponding tests for the absence of BCC. And as we see a need for a test chapter as the other pharmacopoeias develop their own chapters on 60 on BCC, we do have intention of discussing with other pharmacopoeias and making this a harmonized test chapter. But otherwise, at this time, it is a local national chapter for the test for BCC. The next slide. Thank you. So this is these are the questions that were specifically addressed to us on method suitability and also on the types of products that the recently introduced Chapter 60 is going to be applied to. And I hope I answered these questions, but I'll be glad to answer any other questions that you may have. I don't know if we take questions now or at the end of the webinar, but in any case, I'm prepared to answer your questions. So back to you, Kit, and I'll wait for your suggestions, Kit and Amy.
Thank you so much for that. And I'm going to go ahead and pass it over to Shannon now. And I just want to remind everyone that if you do have any questions, please feel free to type those into the Q&A chat. Once Shannon has completed her presentation and we close out here with Kit, we'll have time to address a few more questions. So Shannon, I will pass it over to you.
All right, thank you, Amy, and thank you, Kit and Radha. That was a great foundation explanation on the USP and USP general chapters related to microbiology. So as Amy mentioned, my name is Shannon Jacoby. I'm the business unit manager overseeing the Eurofins Microbiology Lab in Madison, Wisconsin. Previously, we were part of Covance and acquired to Eurofins in 2018. At our lab, we are specialized in specifically probiotics and dietary supplements, and can perform probiotic enumerations by plate count methods, as well as flow cytometry and probiotic identification using PCR or whole genome sequencing, as well as the full suite of USP testing, including the suitability. So there are 4 product categories that we are going to be focusing on today, and why they are applicable to USP testing, as well as some additional guidance on setting specifications and the supporting chapters. So dietary supplements and supplement ingredients can contain highly synthesized components. These components can have unknown antimicrobial properties on contaminating organisms such as E. coli and Salmonella. They can also contain dry botanicals that can have potentially high organic microbial populations, which can also cause competitive inhibition. Additionally, there are several botanicals, or a lot of botanicals, that have known antimicrobial properties, such as cinnamon or ginseng. Probiotics are live, highly concentrated, mainly gram-positive organisms that can also cause competitive inhibition, making other gram-positive pathogens, such as staph and listeria, difficult to recover. Hemp and CBD products are also very similar to dietary supplement and supplement ingredients in the fact that they can either contain highly synthesized components for extracts, or they can also be dry powdered botanicals, also have high potential organic microbial properties, which can cause competitive inhibition. Lastly, personal care products are also highly synthesized, as well as they can contain an antimicrobial preservation. These are utilized more for multi-use containers. Specifically, think of like a lip balm that can pose a risk of contamination by the user. So it uses preservation to reduce any contamination by the user. So why is this important? Well, antimicrobial or competitive inhibition can lead to false negative results. This is why USP requires all product formulations to undergo preparatory or suitability tests to ensure that the resulting contamination testing is accurate. Antimicrobial properties or competitive inhibition of a product could be neutralized at the time of consumption or use, putting the consumer at risk and ultimately putting a company's overall brand at risk. So how does the AOAC FDA BAM methods differ from USP? And they don't, right? So preparatory suitability is still required, but AOAC FDA BAM uses a little bit of a different approach. They tend to use more of a matrix category approach. So it pairs like with like. For example, of meat and poultry has a subcategory of protein, which is less than 10%. This could be a prepared food with high carbohydrates, like a frozen entree. AOAC matrix categories are specific mainly to food and other food types, where dietary supplements, supplement ingredients, cannabis products, and personal care products are difficult to fit into the current matrix categories that are available and would need a new category validation in order to run AOAC FDA BAM methods on these product types. Some real world examples that I've run into in our line of work is we were looking at testing peanut butter, but it didn't align with any of the categories that were listed, but because of the high fat content, aligned mostly with butter. But peanut butter is not a dairy product. We also had an instance where we were testing crickets, and the category that aligned most with was shrimp. So like I said, a new category can be created. It does require some work to be involved, so qualitative methods. There are 3 levels of inoculum, either none, low, and high. Low level should give about 25 to 75% positive tests, and there should be no statistical differences between the new and the reference method. Quantitative methods are a little bit more involved, where they have 4 levels: high, medium, and low inoculum, and none. 5 test portions for each level need to be compared to both the new and the reference method, and the new results are plotted on the y-axis, where the reference method are plotted on the x-axis, and you're looking for discrepancies. And report the mean difference between the new and the reference method and the 95% confidence intervals. USP does have the option to follow the compendium methods using the suitability or preparatory testing. They also have an option to follow 2023, which is the validation of alternative microbiology methods. This is very similar to how AOAC FDA BAM has outlined the new product category. Oh, sorry about that. USP 2023, microbiology attributes of non-sterile nutritional and dietary supplements, offers some guidance on setting specifications. Since setting specifications can be difficult, they need to assess the material origin, in other words, animal, botanical, or is it synthesized? What's the manufacturing process? Is there a kill step? drying, extracting, heat, irradiation, sterilization. What is the contamination potential of the components of the product? Are they growth promoting or are they inhibitory? USP offers guidance in a product's individual monograph, where the monograph provides guidance on a product's definition, identification, performance, impurities, contamination, and additional requirements needed to meet USP standards. If no guidance for contamination is available in the individual monograph, or an individual monograph is unavailable, then Chapter 2023 is used as guidance. For probiotics, if an individual monograph is unavailable, then Chapter 64, probiotics, is used, where they outline probiotic into classification. You can see just a brief outline on the categories and testing guidance. Dried powdered botanicals, powdered botanical extracts, tinctures, fluid extracts, infusions, nutritional supplements with botanicals, botanicals with treated with boiling water before use, dietary supplement ingredients and raw materials, and nutritional supplements with synthetic or highly refined ingredients. You can see here that obviously if a product is more likely to contain organic microbial properties with limited processing, more testing is suggested versus the more highly synthesized products where less testing is suggested. Chapter 64, probiotic, provides testing guidance split by classification of the organisms. There's non-spore forming bacteria. This is going to be your lactic acid bacteria, bifidobacteria, etc. Provides guidance on PCR identification testing and enumeration testing guidelines. Spore forming bacteria, which will be your bacillus species bacteria, and then yeast and molds, Saccharomyces, and so on. In terms of guidance on contamination, non-lactic acid bacteria count by ISO is applicable to non-spore forming bacteria. Yeast and mold contamination is applicable to non-spore forming bacteria and spore forming bacteria. Total aerobic plate count is only applicable to yeast and mold probiotics. E. coli and salmonella are applicable to all probiotic classifications. Additionally, Listeria monocytogenes, Staphylococcus aureus, or Pseudomonas aeruginosa are applicable if any ingredient in the product has been identified as a risk by way of a formal program, such as HACCP. Clostridium perfringens and Chronobacker sakazakii should be tested if a probiotic product is intended specifically for infant use. All of these additional methods listed must be officially accepted methods or appropriately validated methods. So we touched briefly on personal care products and utilizing preservations to protect multi-use products like lip balm. Chapter 51 provides guidance to confirm a product's preservative efficacy. USP 51 provides information regarding a product to reduce and control the contamination population of specific microorganisms identified in the USP. It can also be referenced as challenge testing. What it is not is an efficacy of sanitizing products. It is not a minimum inhibitory test of antibiotics, nor is it a test of shelf life of the product. USP outlines the product types into categories. Category 1s being injections, parenterals, including emulsions, over-the-counter drugs, sterile nasal products, ophthalmics, eye products made with aqueous bases or vehicles. Category 2, topically used products, again, such as lip balm made with aqueous bases or vehicles. Non-sterile nasal products or emulsions, including those applied to mucous membranes. Category 3, oral products other than antacids. And category 4, antacids. Some of the test specifics, samples are inoculated with the following organisms at a concentration specific to the product category. Candida albicans, Aspergillus braziliensis, Escherichia coli, Pseudomonas aeruginosa, and Staphylococcus aureus. Samples are tested at defined intervals based on the product category, either 7, 14, and 28 days for the viable populations of each inoculated organism, and must meet USP's specified acceptance criteria. Generally, it requires a decrease of the viable population, but no increase of the viable population. Just some contact information. Back to Amy.
Thank you so much for that presentation, Shannon. And I'm just going to pass it back over to Kit to give us the overview on what is new at USP. All right, Kit, I just passed it back over to you.
All right, well, let's see if this works this time. Can you see the slides?
Yes, I can.
Okay. So I was asked to just spend a few minutes on what else is going on at USP related to dietary supplements, not necessarily related to microbiology. So I'm going to talk about ongoing activities in actually its standard development. And of course, no presentation these days would be complete without discussing a little bit about cannabis. So USP, the Dietary Supplements Group, has been very busy working on some general chapters. We have a revised chapter 2750, which really helps you comply with the GMPs for dietary supplements. And this is now a chapter only for the finished product dietary supplements. We are now, in PF, having the manufacturing practices for dietary ingredients. And this is a new chapter. It was removed from 2750 and expanded. One of the other big areas that we're working on right now is an area that is a hot topic in botanicals, which are the pyrrolizidine alkaloids. Pyrrolizidine alkaloids can contaminate botanical products because they are found in weeds and other plants that may be co-harvested, and they are extremely highly toxic. So we are working on an informational chapter, and we expect to also come out with a test method chapter on the pyrrolizidine alkaloids. One of the other general chapters that we're working on, we cannot keep up with the speed at which industry creates new combinations and new products. So in order to help support the quality of multi-ingredient products on the market, we are planning a general chapter to help people develop the appropriate quality tests for these multi-ingredient products. This would not include things like multivitamins and minerals, which are well established and have upper tolerable intake limits, but would be for other dietary supplement combination products. We are also working in a lot of areas, but some of the bigger ones, probiotics. We have a lot of monograph work. We're working on a manuscript comparing enumeration methods and also on doing an in-depth safety evaluation of probiotic strains. Proteins, botanicals, we're finishing up some work on cranberry and starting a major elderberry product. Vitamins and minerals, fats and oils, and small molecules. And last but not least, we are working on cannabis. We published in the Journal of Natural Products on medical cannabis, and that has a lot of information about different chemotypes, methods for testing THC and CBD, very useful paper. We are currently developing quality attributes for cannabis-derived products, which would include cannabis extract, hemp extract, and CBD. And that is consistent with the USP resolution number 10, supporting USP in leveraging scientific expertise to develop scientific resources for CBD. We do not take any difference in our compendial processes and admission criteria related to cannabis and its derived compounds. And at this point, I think all of us would be glad to answer questions, and we thank you for your attention.
Thank you so much for that. I've seen a few questions that have come into the section here. So I will open them up and I invite everyone to continue to submitting those questions. I also have included a poll on the screen, so take a moment to respond to that. But the first question that came in today is, why does the USP recommend an ISO method for testing probiotic products instead of testing via the normal USP methods for aerobic plate count?
That's a great question, Amy. This is Shannon. I can answer that. So in terms of probiotics, when it comes to lactic acid bacteria, specifically the non-spore forming bacteria, the total aerobic microbial count test is going to interfere. That method is going to allow proliferation of those organisms. The ISO method has been around testing dairy products for contamination, and a lot of dairy products are fermented with lactic acid bacteria. So the ingredients in the ISO method reduce the proliferation of those types of bacteria, whereas the total aerobic microbial count does not.
Great, thank you for that response, Shannon. The next question that came in states, my current lab uses AOAC BAM methods to test the dietary supplement products that my company produces. Based on the information provided regarding validation of different matrices, are the results for that testing not valid?
Yeah, I can answer that one again. Not necessarily. I would say in that particular instance, you would want to check with your laboratory and confirm what type of validation work might have been done in preparation of those specific products. And then if it hasn't been done, look at performing a project to confirm what the potential for inhibitory properties of the product are, and then making that assessment.
Okay, great, thank you. And I think that I'm going through the questions. We have time for probably one more. This other one that came in, may dietary supplements still be tested under USP 61 and 62?
Can you hear me?
Yes, we can, thank you.
Yeah. So unless there is no specific monograph that says for a dietary supplement that clearly says 2021 and 2022, you can use 61 and 62. It's not that you cannot use 61 and 62, but again, you have to do the suitability testing. And yes, and as I told you, 61, 62 has more rigid requirements versus 2021 and 2022.
Great, thank you for that answer. And we're just wrapping up on the end here. So there's just a couple other points that I wanted to mention. I wanted to remind everyone that a recording of the webinar along with a copy of the slides will be available within 3 business days. So again, if there's any other questions that you have, feel free to reach out to us. But again, we will be sharing that recording of the webinar along with the slides. And I want to once again thank you again for attending today, and thank you to our presenters, a very informative webinar with us.
Thank you for making the arrangements for the webinar, and I thank everyone for their interest.
Thank you.
Likewise here. Thank you very much, Amy. Thanks, Kit, for the opportunity, and thanks to everyone for listening to the seminar. And again, yeah, thank you very much.
Thank you so much.